Take it away, Anne.
Great, thank you. So it's a great pleasure to introduce Elinor Karlsson. She is an associate professor in Bioinformatics and Integrative Biology at the University of Massachusetts Medical School and director of the Vertebrate Genomics, at the Broad Institute of MIT and Harvard. Her research combines new technology, citizen science, and the power of evolution to investigate genome function. Dr. Karlsson's projects include Zoonomia, formerly the 200 Mammals Genome Project, which is an international effort to compare the genomes of over 240 mammals, ranging from the African yellow-spotted rock hyrax, very cute, to the woodland dormouse, also cute, to identify critically important segments of DNA. And in collaboration with Zoo New England, she's developing tools that use genomics to tackle heritable diseases like heart disease, which are common in some zoo populations. So this is known as zoonomics. She has a special interest in dog genetics. I'm getting ahead of myself, although she herself has cats. And her international Darwin's Ark project, darwinsark.org, invites all dog owners to enroll their dogs in an open data research project exploring the genetic basis of dog behavior, as well as diseases such as compulsive disorders, food allergies, and cancers. So those of you who are dog owners can participate. Elinor received her BA in biochemistry and cell biology and her BFA, or fine arts, from Rice University, and earned a PhD in bioinformatics from Boston University. She was a postdoctoral fellow at Harvard University before starting her research group at the University of Massachusetts Medical School in 2014. So welcome and thank you very much for speaking.
Thank you so much, Anne, for the wonderful introduction. Just let me get my screen sharing going here because that's always the big moment of stress, you know, if it doesn't work. Here we go. Can everybody see my beautiful coronavirus? Hopefully. Great. Okay, so thank you so much for the wonderful introduction. I'm really excited to be here. I'm going to talk about the project I, somewhat surprisingly got thrown into coincidentally last March with the new interesting coronaviruses that I hadn't had before, but much more from the host side than from the pathogen side. But I first just wanted to say that hold on a second, let me just get this working. I have this, there we go. Okay, so I'm really excited to be here. Anne mentioned our Zoonomia project, which is looking at comparative genomics across 240 mammals, for which we made 130 new mammalian genomes. And I spend a lot of time at my institutes of human medicine explaining to people that other species do really interesting things that we should probably be interested in terms of human medicine. And so it's great to kind of find out there's a community of people interested in this kind of evolutionary medicine perspective on things. I'm one of many different sequencing consortia out there that are trying to make lots and lots and lots of genomes. You know, there's Zoonomia is our project, there's Bat1K, which just published 6 really nice genomes in Nature, Earth Biogenome, Vertebrate Genomics Project, and a bunch of others. And this is where we got the genomes that we used for this project. But basically, the context on this project I'm going to talk about today is that way back in March, which was kind of the endless month, Harris Lewin, who is kind of heads up the Earth Biogenome Project, contacted me and said that he and his postdoc Joanna had this idea about whether we could use genomics to figure out something about susceptibility to SARS-CoV-2, the virus that causes COVID-19. For those of you who don't know, which probably most people do at this point in time, this is an image of SARS-CoV-2, the virus, a representation of it. Those red things hanging out on its surface are a protein called spike protein. Spike protein is the protein that actually binds to the receptor on the host cells in humans, which is a gene made by a gene called ACE2. And it's that match that allows the coronavirus to infect our cells. Different coronaviruses will use different receptors. MERS-CoV-2 uses a different gene called DPB4, but this is the one that SARS-CoV-2 uses. And it does appear that SARS-CoV-2 is optimized for binding to the human version of ACE2. It's a very nice match. It binds very well. But ACE2 itself as a gene is extremely highly conserved across vertebrates. It does some really, really important things, especially in terms of the renin-angiotensin system. And if you actually look for variation in it, there's not a whole lot within humans or across species. It's just a very highly conserved, very important gene. And this has nothing to do with the fact that viruses are exploiting it. It's just it's kind of base level function. And because SARS-CoV-2 is a zoonotic disease, comparative genomics is really relevant to it. There's several different questions that we were really interested in. Apparently I sounded like I said Siri. Okay, so basically, the first question is, where did SARS-CoV-2 come from? This has not really been answered yet. We're almost positive it probably jumped from some kind of bat species 40 to 70 years ago. There may have been a proximal host, or it might have been a different bat, or something went on. And then much more recently, it jumped into humans. Which species are susceptible to infection with SARS-CoV-2 is another one that's really kind of topical right now. Shortly after the outbreak started, we started realizing that humans were not the only ones getting infected. There's stories out about the Bronx Zoo tigers getting sick. They were infected by their caretakers. The Dutch mink farms had massive outbreaks. There's cats that have been infected by it, et cetera. So we knew that it wasn't just humans that were susceptible to this. And then there's also the problem of viral reservoirs. Is it possible for this virus to get into another species and actually set up kind of its home there and actually continually spill back into the human population, which could mean that we have an ongoing challenge, even if we control it in the human population. And this is what you see for MERS coronavirus. It's not actually particularly transmissible among humans, but you have constant spillover events from camels back into humans. And so the question that Harris had approached me with, and several other people, was basically, can we use existing genomic data sets? Entirely existing, because by this point in time, it was March, we were all stuck at home, nobody could go into the lab, but there's a huge amount of data in there and databases out there. So could we use that existing data to make some kind of prediction about which species were likely to be susceptible? And we got this fantastic collaboration of people together. Oh, and I just wanted to address the reason why we were doing this. And the reason why it's so sensitive. There's obviously lots of laboratory methods for actually looking at susceptibility, but you can't do that on a whole lot of species, and it takes a lot of time, and also the labs were shut down. And at the same time, because of stories like the tigers getting sick, both the zoo community and the biodiversity conservation community was reaching out to anybody who knew anything, trying to figure out which species were at risk of this disease, particularly for the ones that were endangered and could actually really, possibly be at risk of extinction if they had major outbreaks. And then also human, we knew that mice weren't susceptible, but we didn't know what model systems could be used for therapeutic development. And so we put together this really fantastic group of people kind of from that whole world of genomic people sequencing genomes that I talked about, as well as some protein people and things like that, and started looking at what we could actually do with the data that we had. And we went into the databases and we got to talk to people that we knew and just try to figure out all the species we could find out there. And we ended up with a data set of 252 mammals, 72 birds, 65 fishes, 17 reptiles, and 4 amphibians that we had an ACE2 protein coding sequence for, and we lined them all up and basically made a 410-way alignment of all of these species. And the question we asked was actually very simple. We compared the host receptor of ACE2, so the sequence of ACE2 in each of these 410 species, and asked which was the best fit to the spike protein on SARS-CoV-2. And the way we are defining best fit here is basically how much it looks like the human ACE2 bound to SARS-CoV-2, because by this point in time, we are very, very, very certain that humans are extremely susceptible to this virus. And so we know that that is a really good close relationship between those two proteins. And so this is the paper we published. We actually got into bioRxiv within a month of when I first got that email from Harris. It was one of the most interesting paper writing experiences that I've had. It's quite stressful. You're trying to get it out quickly because it's very topical information and you want to get it out, but at the same time, you don't want to get it wrong. It was really kind of an interesting experience. And then much more recently, it actually came out in PNAS as an after review and everything. And it came out with this very nice figure that shows all our predictions. So I just wanted to talk about exactly what we were doing here in terms of how we were handling the data. And so there were basically 3 different things we did. The first we did was use the amino acid substitution analysis across all of those species to try and predict which ones, based on that sequence, were likely to bind to the SARS-CoV-2 spike. We then also used a kind of complementary method of homology modeling of the actual protein of ACE2 spike interactions to see whether the substitutions that were in there were likely to disrupt those interactions. And then finally, we did a basic kind of selection scan to see whether we could actually detect any evidence of positive selection on residues in the ACE2 binding domain in different mammalian clades. We were actually really helped by this because obviously there was a huge community of scientists at that point in time that were studying this virus, and 2 groups had already published crystal structures for the relationship between SARS-CoV-2 and the spike protein, and the human ACE2, which was actually really helpful. So we weren't relying on the models for older viruses. We identified 25 amino acids that were in the binding site between SARS-CoV-2 and spike that we used in our scoring scheme that we came up with. And we basically, as we often do in genomics, came up with a way to rank the different species. There were several residues in the binding pocket that we knew were really important based on older work done with SARS-CoV, not SARS-CoV-2. And so we kind of made those more important in the scoring scheme. And so it's basically a combination of how much are your amino acids matched to humans, and then how many of the really important residues have changes in them. And we had a 5-category system from very high, high, medium, low, and then very low. And the first thing we noticed was that everything that wasn't a mammal was low or very low, though none of those seemed to be particularly susceptible to this disease. As I said, we paired this with homology modeling. This was work done by Corey Painter and Nicole Persky at the Broad, who had lots of experience doing this, which was a lot. It was actually really fun because as genomics people, many of us hadn't thought about protein structures in a while, so it was actually really interesting to work with them. And we took, as I said, the positions that were changing and tried to predict which ones would actually not be good in terms of that relationship between ACE2 and the SARS-CoV-2 spike. And that's just graphed down at the bottom. So basically, and if you're in the lower left corner, you don't have very many unfavorable interactions. And if you're in the upper right corner, you have lots and lots of unfavorable interactions. And what you can see is that the homology modeling was tracking pretty well with what we'd scored the species as on our amino acid analysis scoring scheme. All of the high-risk species were primates, and specifically old world primates and things close to humans, and they all looked exactly like humans, and our prediction would be that they're all incredibly susceptible. Things got a little bit more interesting in the next category down, which was risk. And here you can see that we still have some primates, but we get a lot of cetaceans and rodents and things. And so that's was actually quite interesting in terms of how it broke down. What we saw was that, as I said, lots of primates in the very high risk group, which is red here. But cetaceans were mostly in the high risk group, which was kind of interesting. We don't know whether they're susceptible or not, but it could be something that should be looked at. And then the other thing we saw, of course, is that the bats, Chiroptera, actually none of them were anything. We had lows and very lows, which is the darker blue, and nothing higher than that. And if we actually looked back at what we knew about species and how susceptible they were, so if we take the cats, like the tiger that I talked about, they're actually in the medium group. And it seems like the medium group actually works pretty well in the sense that they tend to be able to get infected, but they don't seem to get that infected, and they don't seem to get that sick, and they clear the virus, and they're not particularly infectious. Dogs were in the low group, and that's consistent with what we've seen so far, is that it doesn't really, they might have detected infections in a couple dogs, but they don't get sick at all. So it's very low level. It might just be that it can bind a little bit, but not really well. And we did look, there's not, as I said, infectivity studies are hard. That's why we were doing the genomics to begin with, but we looked at the data that was out there and basically saw that if we looked at our prediction, which is on the left, and the data that was out there, we saw some really nice kind of correlations for most of the species. It's not a lot, obviously. Hi, kitten. You were in the medium group. We did have one exception, the ferret, which is kind of an interesting one. So this is an example of what we saw. This is the rodents. As I said, we know that both Mus musculus, the house mouse, and the rat are not susceptible to this, the brown rat. And what you actually see here are things like this particular amino acid substitution, which is not seen anywhere else in any of our 410 species. And it actually, what it does is it introduces, it basically loses contact that you see in the wild type, which is the K353. If you have that mutation to a histidine, you basically disrupt that contact. And what we said, they've seen this in cellular assays that mouse ACE2 really doesn't bind SARS-CoV-2. This is a pretty sensitive assay. There's a lot of other species that have no evidence of being susceptible on a kind of organism level, but are susceptible in this kind of cellular assay, which is basically where you take human cells and get them to express the species ACE2. Mustelids, as I said, are strange. They all get rated as low, but they are clearly susceptible to infection. The ferrets don't get that sick. The minks do get really sick. I don't know what's going on there, but it's possible that the ferrets and the minks and the mustelids might be using a different receptor, or they have a different protein that's kind of making them more susceptible. Because as far as we can tell, there's some clashes. There are clashes here, which would suggest that it's not binding between ACE2 in the mustelids and SARS-CoV-2, but it's clear that they are getting infected. And so we're not entirely sure how that's happening. And then the bats. And so bats are really interesting because they're the reservoirs for beta coronaviruses like SARS-CoV-2. They seem to be able to be infected with them and they don't get that sick from them. So they carry them around and the viruses have time to evolve. And it's really kind of an interesting question as to how they do it. How do they actually carry these viruses without getting that sick from them? And so we looked at selection, basically looking at two methods, Philo-P, which is a method for identifying residues that are basically changing faster than you would expect under the neutral rate. So it doesn't really care about, you know, exactly what you're changing to or whether it's functional or not. It's just that it's changing. Hi, and then CodeML, which is actually looking for codons with an excess of non-synonymous mutations. And this was work done by Graham Hughes and Kathleen Keogh. ACE2, as I said, highly conserved in all mammals. That's the green positions here are the highly conserved positions, and you can see that it's most of them. Blue is accelerated, meaning that it's changing faster than you would expect under the neutral model. Very highly conserved. There's a little bit of evidence for selection, but not a ton. So this was just a picture of the few residues that we happened to find that had a signal of accelerated evolution and came up under the CodeML analysis as well. And so the red ones are actually in the binding interface. So there's some stuff there, but it's a little bit hard to know how to interpret that. But what we did see that is a little bit more interesting possibly is that bats do have more accelerated residues than other clades. And so they have more residues that are changing faster than you would expect by random chance compared to other clades. And when you actually look at this, you'll see that there's a whole bunch of these that are actually sitting in the binding interface between spike and SARS-CoV-2. Selection analysis is a little bit hard because obviously we keep on interpreting everything through the lens of does it bind to SARS-CoV-2 spike protein, but that is not the natural function of ACE2, and ACE2 does many other things that would explain why it was under selection, that could explain why it was under selection. That has nothing to do with viruses. But I do find it intriguing that what you're seeing is something different going on in the bats that we know can tolerate these viruses, and that we see this excess of selected residues really right in the binding interface between ACE2 and the spike protein. And there has been some follow-up work in a recent paper that actually looked at the M82 and the T27 position, which are two of the ones that we found in this analysis, and showed that amino acid changes that are carried in the bats tend to decrease binding to SARS-CoV-2. So that's consistent with that. And so I think right now what we're kind of thinking about with this analysis is first, where do we go from here? So we've got some funding from NSF and we're looking at extending this from ACE2 to other genes that are involved in infection. Clearly, the fact that the binding between ACE2 and the spike is just the beginning of infection by SARS-CoV-2. And so there's a lot of other components to that pathway that could be controlling whether you get infected and whether you get sick. We'd like to extend this to non-coding regulatory sequences and not just the coding regions of these genes. And it would be really nice to actually be able to pair it with some experimental assays of infectivity so we can actually validate how well this kind of genomic prediction works. But I think there's also kind of an interesting philosophical question with this kind of analysis, which is that you can always argue that the genomic predictions are just predictions and we don't have the functional data to back it up. And that's very true. And in order to know whether a species is susceptible to something like a new virus, you are going to have to actually do tests and figure out whether they're susceptible or not. But at the same time, we managed to come up with predictions for 410 species, which, while flawed, is a lot more information than we had before that. And for people that are caring for these animals, it may be really valuable information in terms of knowing which ones are at highest risk and that you really need to try and protect. And as I said, particularly for the ones that are endangered, this approach could be particularly valuable, both because they're endangered and you don't want them to die, but also because you can't do a lot of experiments on these animals. It's not like you can go and kill an endangered species in order to do some studies on its tissues particularly easily. And many of the species that we're talking about here will not tolerate captivity. So there's no way to actually do studies on the animals themselves. And the cellular assays are always going to have the same limitations that you kind of talk about with genomics, which is it's not a real world test. So it's kind of been an interesting project for me, just kind of getting into a whole new world of viral kind of susceptibility that I hadn't really thought about much before. And then the last point I wanted to make is that there are a lot of species, even with genomics, that we could not come up with predictions for, because there's just, the genomic data out there is still really limited. And I always like to end with this call out to keep on sequencing things, because we've really only explored a tiny proportion of the animal tree at this point in time. And this is only, for most part, one individual per species, which is just starting it. And especially at places like the places I work, this is just the number of species in each of the group, because we're getting really, really good at sequencing humans over and over and over and over again. And so I think figuring out how we can use comparative approaches to inform the human side of things is a really kind of interesting challenge at this point in time. And then I was just going to end by saying thank you to everybody that worked on this project. As I said, it was a really fun project to do, both from a scientific standpoint and just from a, we're all stuck inside and it's a pandemic and everything in the world is weird, and you actually had a mission all of a sudden and a group of people to work with on it. So it was fun from that point of view as well. So thank you very much. Those were the slides that I had.
That was great, Elinor. Thank you so much.
Oh, you're welcome.
Fantastic. I'm going to suggest, okay, exactly. Stop sharing the screen so we can see each other a little bit better. We have a lot of questions in the chat box, and I'm not as good with Zoom as Meredith is. Do we have any hands raised?
Don't have hands raised at the moment, so feel free to go ahead and virtually raise your hand to prioritize your question from the chat, or if you have a new question that's not in the chat yet. But to get us started, I'd actually like to get Charlie to ask his questions. That's something I'm interested in as well.
Yes, what was my question? What are some of the, you mentioned that there were, probably many selective forces on these ACE2 receptors. I wonder if you could say something more about that on these genes.
I don't know all that much about, ACE2's function, because it seems like it's, like everything in genomics, when you actually go and prod into it, it turns out we know a lot less than we think we do. But I think it's an interesting part to this story from two directions. One is that understanding how selection acts in the context of an infectious disease, it's really easy to get biased by a focus on the infectious disease itself, when infectious diseases are probably the kind of outlier event in this situation. But it's also interesting in terms of both susceptibility to the disease and all of the different side effects that we're seeing. I think I've heard some speculations that part of the reason why this disease is so dangerous is that ACE2 is expressed in nearly every cell type. I mean, especially in things like fat cells and, the linings of all the vascular cell types and everything. And so when you talk about having these really diverse side effects from this disease, or these diverse symptoms, it might be that that's because of how widespread the ACE2 expression is. And it's kind of an interesting question about whether, when you're thinking about infectious diseases, what gene that disease chooses to use as its receptor is actually going to shape the pandemic that you come out with from all of it. So it's also this interesting question of which genes are actually proteins can be used as receptors that we're trying to figure out, because we have this habit in science of only looking at things when they're actually a problem. And can we actually explore more of the space of the potential receptors out there, outside of the ones that we actually already know get used?
Thanks for that. I see that Brandon Ubunu has a question. Brandon, do you want to ask your question?
Yeah, magnificent talk, fascinating. Thank you for sharing that work with us. I mean, I think some people would push back on the notion that that this hasn't been under selection the entire time by viruses, right? So no, right, but that's neither here nor there. I think my question really is much more that what's exciting about your approach is you look at the side of the problem from a direction that people have not. And I wonder if there's any talk between your side of this and this idea of a watch list, a viral watch list, right? Where you're sequencing variants and that there are alleles that are like, I know in Ebola, it was like an escape variants for a binding antibody or something like that. So any kind of notion of talking across the aisle here, so to speak, and kind of getting some kind of matched biophysical modeling, both on the host receptor side and on the viral epitope side and matching that somehow. Does that make sense?
Yeah, no, it does. And I think it's a really interesting idea because evolution, you know, as you see things evolve, you should be able to figure out which things are actually functional, you know, important changes versus not important based on the patterns of evolution. And I think one of the things that I was really struck by when we started digging into this disease was the fact that the reason why, I think there was a question about why we think it was bats and not humans infecting the bats. It's actually an interesting point of view. One, I mean, humans wouldn't start, no, I'm just kidding. So bats are just really interesting because they have a pattern of probably immune suppression that may or may not be related to the fact that they fly. That means that they can get infected with a lot of viruses and not be affected by them, which from the viral standpoint gives it an opportunity for the viruses to evolve kind of in these living hosts, which is kind of an interesting perspective on it. But what really surprised me was that the reason why we think it came from a bat was because some person in some study a couple of years ago in one bat population in one cave happened to sequence a virus that was the closest thing we have found so far to the one that's in humans. But it's not the same virus. And we don't know what the viruses are that are out there. We've looked at so little of the normal viral landscape that's out there in the world that it's impossible to predict. We only pay attention to them once they start killing us. And so we don't have any of the background information to actually compare what we're seeing in the pathogenic ones. And I think one of the things that we're doing on the NSF grant that I'll be really curious to see if it actually works or not, but we propose just going into the data that's out there. So there's the RNA-seq data that everybody's generating from all of these species all over the place. It turns out that coronaviruses are polyadenylated, which means that if a person is infected with the coronavirus, when they're doing the RNA-seq on it, we should also be sequencing the virus itself. And it might have ended up in the short read archive and the sequence read archive because it was in there and everybody probably ignored it because it wasn't the thing that they were actually interested in sequencing. And so we thought, well, let's just go into the SRA and see whether we can find coronavirus reads or not, to see whether we can expand what we know about just normal diversity out there. So to answer your question, I think evolution would be really interesting, but it's hard to look at evolution without having the background landscape.
That's right. Thank you. Thank you.
Great, thanks. So there's a really interesting thread going on in the chat box, initially initiated by Benjamin Auerbach, who had asked about the variation and whether variation was possible in the binding of the spike protein for it to make the virus infectious. And then Anne Laudosiot had responded, or sorry, wrong response. Amanda Mellon had responded that they did work on protein-protein interactions and found results consistent with urinalyses. So Benjamin then asks, how closely do the proteins have to match to bind, and whether there's a way of predicting how the spike has been able to bind differently among taxa given its current morphology in humans, and then whether that could be used to clue us into potential intermediary species. Do you have any thoughts on that?
I can tell you that we had a lot of discussions when we were writing the paper as to people proposing that there's absolutely no way that that thing could actually still bind to that ACE2, and they must be using a different receptor to, yes, it probably binds sometimes and it's just close enough that sometimes it can get in. We don't have an answer to this, but I think it's a really intriguing question as to whether you could actually predict binding with enough accuracy to be able to tell whether a species can use ACE, whether the virus can actually use that species ACE2 in some kind of much more defined way than what we came up with, which is basically it looks better or it looks worse. I'm not a protein structure person, so I will defer to anybody who's actually a protein structure person to talk about that. But I think it would be really nice to be able to get the genomics. I mean, one of the things that was interesting about this paper is that Corey and Nikki got involved. One of those random things that happens in science, because I was talking to Corey about something entirely different, and then she put up a tweet about protein structure and how she'd done it during her PhD. And I was like, you know something about protein structure? We have this project where we're looking at ACE2 and SARS-CoV-2, and she got involved. And it was both interesting and challenging at times, because it turns out that the protein structure people and the genomics people are talking totally different languages half the time, and we had to figure out where we actually overlapped. But when we could make it work, it was really useful. So
great, thank you for that. So it looks like we have a question from Kayla Kaufman in the chat. Kayla asked, do you have any preliminary findings on how the non-coding regulatory regions compare between species that we know are susceptible but don't show clear susceptibility in the coding regions?
Not yet, but we'd like to look at that. It's a bit of a chicken and an egg problem, because when you get into the non-coding regions, it's much harder to predict function, obviously. And so when you combine that uncertainty about whether a change is actually going to be functional or not, combined with, for the vast majority of species, having no good data on whether they're susceptible or not, it ends up with a lot of unknowns on both sides of the equation. And it's much harder to look at function in a regulatory region in a single species or even a handful of species. It would be nice to have a bigger data set than that. One of the reasons, so the actual motivating principle behind the zoonomia project that I mentioned at the beginning, and the reason why we made an alignment of 240 eutherian mammals, was that we wanted to identify positions in the genome that were exceptionally conserved. So we know that only somewhere between 7 and 10% of the human genome is actually functionally, the sequence is actually relevant, is functionally important. And we wanted to identify exactly which positions in the genome were those functionally conserved positions. And so we've thought about trying to kind of look at whether something is exceptionally conserved across all of the mammals, but changing in the subset of lineages where we do have susceptibility information, but we haven't done that yet.
Great, thank you. We also had a question in the chat from Sudhir Kumar, wondering if you've used your model to predict individuals susceptible to COVID-19.
No, we do have in the PNAS paper a section on variation in humans. We did do as much as we could a dive into what variation you see in human populations in the binding interface between SARS-CoV-2 and spike, and the answer is basically you don't. So there wasn't much we could do with that. There's very little variation in humans, and it didn't seem like any of it was likely to be functional. And so it doesn't, my prediction would be that variation in spike in the SARS-CoV-2 spike, the ACE2 spike binding pocket in humans is unlikely to be a major determinant of whether you're susceptible or not.
Great. So it looks like we had a question from Charlie. Charlie, you can go ahead with your question.
Sure. I can ask maybe a question from a slightly different thread. There was another, I've been hearing about this other human coronavirus, an HCOV, from like the mid-19th century, that I think it was a spillover from cows. And I've just been really curious about that. And I wonder if you know anything about the history of that, its impact on humans, any kind of coevolutionary responses, the virus and the cattle, or some and the human, and or maybe somebody else here knows about that history. I've just been fascinated about it, and I just wonder what's known about it. I haven't looked up anything on it.
Yeah, I don't know anything more about it than that either. I think it was something that I discovered because one of the questions that we were interested in was this whole possible reservoir species question. And so the fact that many different species have been, I shouldn't say many, the idea that different mammals have been shown to be hosts for coronaviruses in humans is relevant to the question of what could be a host in the future. And so, when we looked at it, cattle were not something that came up as being at risk. Pigs were not something that came up being at risk, but reindeer and white-tailed deer were. And so it was kind of like, could one of those be potential reservoirs in the future kind of question. But if anybody does know anything about the 19th century one, I don't, so.
Yeah, does anyone else know anything about that and want to share with us about it, its impact on humans and any of the evidence for its origins?
If you do, feel free to raise your hand. We can always return back to that topic. But it looks like we do have a question next from Evan Gehring. Evan, you can go ahead with your question.
Hi, great talk. I was wondering if there's any evidence from lab experiments or comparative studies that host shifts to species with different ACE2 receptors lead to evolution in the spike protein.
So I guess the answer would have to be yes, in a bigger sense, because we know that somehow whatever's infecting us now is really well adapted to humans. And that happened presumably through several hosts away from whatever as I said, the only thing we have right now is that data point from that virus that somebody happened to sequence out of a bat. I think the assumption would be that being a virus, what it's doing is adapting to, it gets into a new host and then it makes itself at home, basically. And I'm not an expert on this, but my understanding right now is that, it's one of these slightly confusing things about this virus and the timing of it, because it is so well matched to humans. But at the same time, within the space of the pandemic, it's not changing very much. It's actually a pretty stable virus. It doesn't seem to be undergoing any kind of rapid evolution. And so the question is, how did it get to be so well matched to humans? And I don't know the answer to that. And I'm sure there's people that on the viral evolution side that have all sorts of theories about that. But I think it's I haven't totally figured out how to make that timing work in my head. Like, when did it get so well matched to humans and how did that happen if it emerged recently and it's not changing very quickly? So.
Thanks for that response. So it looks like next we have a question from Ayana Paul. Ayana, you can go ahead with your question.
Hi, I just had a question on how you and the other researchers came up with the classification categories for the ACE2 and SARS-CoV-2 binding ability prediction. Like, how did you look at the amino acid sequences first and then decide what was considered a very high binding ability and what was a low ability? I just wanted to know, like, how.
Yeah, no, it's a great question. So it was a little, to be totally honest, it's a bit ad hoc, as many of these kinds of classification schemes are, in the sense that we needed to come up with some way to rank things. And the most straightforward way to do it, and the way that we tried it first, was just to simply do it based on identity to humans. So the more amino acids you had that matched to humans, the closer you were. But even based on the few data points that we had, it was pretty clear that we were missing a lot of information just going based on simple identity, because we did have some information out there on which positions were the most important. And specifically, there'd been a lot of work done on SARS-CoV, not SARS-CoV-2, but SARS-CoV, which is the one that causes SARS, which was the big, the big spillover kind of event in, I guess it was about 15 years ago now, and they managed to get control over that one. But subsequent to that, people did a lot of research looking at exactly, using mutagenesis experiments to change positions in that binding pocket and show which ones actually disrupted the relationship between SARS-CoV and ACE2, because SARS-CoV also used ACE2 as a receptor. And so what we basically did is we took the information from the crystal structures of SARS-CoV-2 and ACE2 to identify the set of 26 residues that we wanted to look at. But then within that, we identified a subset, I think of about 7 or 8, where the mutagenesis experiments using SARS-CoV had actually shown that changing those positions was especially important. And so we kind of combined the two approaches. And that basically meant that we fell out into about, we could actually have a 5-level scheme where we had some that were coming out, we kind of had separation between the very high ones and the high ones. And we seemed to see something that was useful there. And then it matched up fairly well with what was coming out of the kind of infection susceptibility literature. But to be totally honest, it was a bit, genomics kind of figure it out as you go, because there's not a lot of information out there to base it on. So that was kind of part of the reason for combining it with the structural homology part too, was that it just provided a little bit more information that what we were looking at was actually likely to have actual repercussions, and it wasn't just a scoring scheme based on amino acid identity.
Thank you.
Explaining that. So it looks like we have Amanda Mellon who wanted to share some perspectives. Amanda, you can go ahead.
Thanks. Yeah, it's Malene. It's a tricky last name. I just received a request to perhaps share a little bit of insight from our study. So we'd taken a very similar approach. I shared the link to bioRxiv through the chat room, but it should be out in communications biology maybe next week. And our group took a similar approach, but we only focused on primates. And we found some considerable variation in lemurs, and also in the platyrrhine, so the monkeys in Central and South America that looked a lot different from the catarrhines that were discussed here. We were quite concerned about how susceptible they would be from forming kinds of conservation efforts and captive management protocols. And so in addition to doing the modeling from genomics, we also worked with Bobby Ora, who's a professor at NYU who does protein-protein interaction modeling. And they're able to use a technique called alanine scanning, where you look at those critical binding sites and you can evaluate each one's contribution to the binding complex between ACE2 and the SARS-CoV-2. You change each one of them by replacing it to alanine, and you can look at the relative strength and the interaction. And by looking at the specific sites that differ between the platyrrhine primates and those that are found in Africa and Asia, look like about a 400-fold reduced binding affinity, which is also backed up by the infection studies. The results presented here today, we see that macaques and vervets can be really easily infected with human-like symptoms, but when it's examined in marmosets, they still have some microlesions, but far lower responses. Unfortunately, it looks like some lemurs based on these models might be quite susceptible, but the next step in this type of research would actually be to actually express the ACE2 protein. You can do site-directed mutagenesis to change different bits of it, and then get some synthetic SARS-CoV-2 spike binding domain and test experimentally some of these predictions. So the infection studies are backing up by the protein-protein modelings, but basically, looking at that way of analyzing these data is strongly backing up the types of analyses that Elinor was presenting today.
Thank you so much, and that's fantastic to hear. It's one of those things when you do these kind of genome scale big species things, is that, you know, scientifically it could be slightly unsatisfying because you come up with these predictions, but it's just a prediction, right? And it's like you want to actually know what's going on. And so it's really cool to actually hear that, one, you were able to take a deep dive into the primates, because obviously a bunch of those are the really high concern species. And just telling people that they're all at risk is less useful than actually being able to figure out what the critical factors are and which ones, you know, seem to be the kind of highest ones. But also just what a lot of work. That sounds amazing. Like, I'm really looking forward to seeing the paper.
Great. Thanks so much, Amanda, for sharing that. Looks like we had a question from Dawn Stansel in the chat about the timeline of the study. Seems like it all came together very quickly. Obviously, it was a time-sensitive project. So Dawn was wondering exactly how much time passed from the start of this to publishing. And she notes that it's very impressive how it all came together.
Impressive and scary as well. So I checked the dates, as I said, when I wrote a little Twitter story about the paper when it came out, which I found is quite a lot of fun. And between when I got the first email from Harris Lewin saying, hey, I've been thinking about using genomics, and when we put the paper into bioRxiv, it was less than a month. And it was, you know, there's something about a pandemic that can, you know, everybody always wants to be open and sharing and collaborative in their science, but because it was a pandemic, everybody was, to a degree that you almost never see in longer projects, where it was everybody just kind of went all in without having a lot of, you know, this is the person doing the most and this is the person doing the least. And it was really the only way that we could have done it that quickly. Because especially, you know, in terms of the science part of it, it's stressful, because often it's those iterative processes that take a while is where you make sure that everything you're doing is right. And the idea of putting something out that quickly, you start getting really worried that something that you've put in there is not right. And so having many different people poking at it constantly. Oh, also, I recommend having many different time zones. That helps a lot too. So we had some people in Europe and on the West Coast and here. And so we actually managed to cover most of the 24 hours in the day. Plus, none of us had anywhere to go because we were like, you know, I was in Boston at the time, was totally locked down. So we were all stuck at home. So.
And how about dealing with the press? Have you gotten press attention for this? And you know, I noticed in your paper you were cautious in terms of the significance statement, in terms of, you know, what could really be said. I just wonder how you've navigated, you know, communicating this to the public, if you've been doing that at all.
Yeah, there was actually a lot of thought that went into that. And one of the great things about this paper that you'll notice if you notice the author list is that we had people involved both from the Smithsonian and from the San Diego Zoo. So Ollie Ryder is at the San Diego Zoo. And Harris actually worked with the zoo on figuring out the press statement to put out, you know, the actual press release, to make sure that it kind of covered that side of things from a very responsible standpoint. And they also did several presentations to that community well before the paper came out, right from the beginning. Because as I said, part of the thing that motivated it is that they were getting questions on a daily basis about which animals were most at risk. And so as soon as we started getting things out that seemed reliable, we started talking to that community about it, just to, you know, to try to get the information out as quickly as possible. But there is, I mean, the biggest problem is probably the bats and how you talk about them, because you know, the last thing you want to do is demonize the bats. And so figuring out how to make sure that that's not what you're doing when you talk about this, I think is probably the thing I was most worried about. And it seems to have been okay. I haven't had any issues so far anyway.
Thank you so much, Elinor. This was fascinating.
Thank you for inviting me. I had a lot of fun. This was great. And thank you for all the fantastic questions.
Yeah, thanks, Elinor. This is a great talk, and thank you all for joining. And Anne, thank you for doing the introduction and for nominating Elinor as a speaker in Club EvMed from Center for Evolutionary Medicine, a nomination. I want to just share quickly our upcoming program, if I can do this. You should be seeing our webpage for Club EvMed and today's talk. And if you scroll down, you will see that next week we have Brian Hare speaking about his book. It will be another book episode of Club EvMed. His book is titled Survival of the Friendliest. And that will also be on Tuesday. As a lot of you know, these are not regularly on one particular day. They can vary depending on when the speaker is available. And then after that, we have a talk that's cancer oriented with James D. Grigori. I hope I'm pronouncing that correctly, on Monday at 1 o'clock Eastern time. And after that, we have Tobias Lenz speaking with us October 27th on a Tuesday, 1 o'clock Eastern time. And you can see that we have more in the works, and we're already lining up talks for next semester, for next winter. So please stay tuned. So with that, we're going to close this episode of Club EvMed. Thank you guys so much for joining. Thanks for the fantastic conversation and perspectives that are shared in the chat box, and hope to see you guys next week. Thanks again, Elinor.
Thank you.